Assalamualaikum and good morning everyone. Okay, previously in our previous lesson you have learned about gene expression. Okay, genes, they are being expressed via transcription and translation. Okay, so this gene expression process, or should I say the transcription and translation, okay, of gene, okay, it needs to be regulated. Okay, perlu dikawal. Okay, so that brings us to regulation of gene expression. Okay, now gene expresses itself through protein synthesis involving transcription and translation. All right, you have learned this already. Okay, and then gene expression is affected by the environment. Okay, example, development of the human hemoglobin, whereas fetal hemoglobin, okay, as you have known, it has higher oxygen affinity compared to the adult's hemoglobin, because the fetal hemoglobin consists of the alpha polypeptide and the gamma polypeptide. Okay, so what happens just before birth, the synthesis of the gamma polypeptide stops, and this activates the synthesis of the beta polypeptides. That's why adults, or sorry, young children or children, especially babies, okay, and throughout adulthood, we have only the alpha and beta polypeptides. Okay, that's about gene expression. Okay, now let's take a look at deeper part of this regulation of gene expression. Okay, now regulation of gene expression, okay, it involves the operon system. Okay, operon system. This operon system can be found in the DNA of prokaryotes as well as eukaryotes. Now, the thing is, this operon system, how they really truly functions in the eukaryotes are not yet properly understood even until today. Okay, so what you are going to study is about the operon system or regulation of gene expression that occurs in the DNA of a prokaryote. Okay, one example of prokaryotes that shows that exhibits simple regulation of gene expression is E. coli. Okay, through the lac operon and trip operon. But for your syllabus, you are only going to study about lac operon. Right now, what is this operon system? It is a system consists of a regulatory gene and an operon, okay, that control or regulate the protein synthesis in prokaryotes. So this is, okay, wait, pointer. Okay, so this is an operon, right? And sorry, an operon system. And this operon system here, it has the regulatory gene over here, is what you can see. Okay, and the operon. All right. Okay, now what is an operon? It is a functional unit that consists of genes, okay, arranged in a continuous array and consists of promoter. Okay, this is the promoter of an operon. Okay, this promoter here, it has a small part of it, right, which is known as the operator. Now, what is the promoter? It is the site of attachment for RNA polymerase to start transcription by the structural genes. This is the structural genes, like Z, like Y, and like A. Okay, now what is the operator? Operator is a part of the promoter. It activates or deactivates transcription of structural genes. And this operator also serves as binding site for the repressor protein. Okay, I will show you later how this repressor protein looks like. Okay, so for operator, it has two functions. It's a binding site for the repressor protein, and it controls the transcription of structural genes. Now, what is structural genes? Okay, they are genes with enzyme coding sequence for transcription, or you can simply say that structural genes are genes that codes protein from transcription. So genes with enzyme coding sequence for transcription. All structural genes in an operon will be transcribed as a single transcription unit. So meaning to say, during transcription of structural genes, all the genes in the structural gene will be transcribed as a single transcription unit. There's no such thing as when the RNA polymerase passed through this structural gene, it only transcribed like Z, for example, only like Z. Sorry, only like Z. No, it will transcribe like Z, like Y, like A altogether, simultaneously, concurrently, as a single transcription unit. A regulatory gene, as what you can see from here. This I here. Regulatory gene. It's a gene that turns on or off the transcription of other genes. So it's a gene that turns on and off the transcription of other genes. Other genes is being referred to the structural genes. So say regulatory gene, it either activates or deactivates the transcription of the structural genes. Now, one example of regulatory gene is the LAC-I gene. LAC-I. Now, class, I need to highlight here. This LAC-I, small letter L, A, C, and then capital letter of I. And apparently this LAC-I, they are being when you write LAC-I, you have to underline them. Now, LAC-I gene, as you have known, is the regulatory gene. It is not a part of the operon. This is the operon. Remember, I told you this is the operon. It's not a part of the operon, and it has genetic information for repressor protein synthesis. It has genetic information for repressor protein synthesis, meaning to say transcription and translation of this regulatory gene will give or will synthesize or will produce a repressor protein. So this is transcription process, which will give you a mRNA, and then translation of this mRNA will give you a repressor, and this repressor is always active, right? So an active repressor will bind to the operator to inhibit transcription of the structural genes. That's why a regulatory gene is a gene that turns on or off the transcription of other genes. A repressor protein example is the LAC repressor, because we are now talking about LAC operon, because LAC operon is the operon that is being mentioned in your syllabus that you must know, right? So a LAC repressor, a LAC repressor is a repressor protein, and it is a negative regulatory protein because it inhibits transcription when bound to DNA. Bound to DNA refer to the operator site, because this operator site is in a DNA. That's why LAC repressor, or the repressor protein, is a negative regulatory protein because it inhibits transcription when it is bind to the DNA. Synthesized Okay, this lac repressor is synthesized from the lac-I gene and binds to the operator to block the binding of RNA polymerase to the promoter. This inhibits transcription of the structural genes. So apparently, okay, class, this RNA polymerase, when it binds to the promoter, okay, and if this repressor here is not on the operator side, what happens? This RNA polymerase will pass through the structural genes and activate the transcription of the structural genes. Now, repressible enzymes. Now we are familiarizing with terms first, okay, the terms. Then we proceed with the regulation process. So repressible enzymes are enzymes that are synthesized consistently, regardless the presence or absence of certain substrate, okay, substrates or substances. So one example of repressible enzymes is the tryptophan synthetase. Okay, and these repressible enzymes, okay, or tryptophan synthetase, is being shown by the trip operon. And this trip operon, TRP operon, is not in your syllabus. Inducible enzymes are enzymes that are produced in the presence of specific inducer molecule. So meaning to say, okay, in inducible enzymes, these enzymes are always being synthesized. They are enzymes, okay, certain enzymes that are always being synthesized, provided, okay, there is a substrate, okay, and then the substrate is known as inducer molecule. Now for the lac operon, okay, the enzymes that are being synthesized are beta-galactosidase, permease, and transacetylase. Now, an inducer, as what I mentioned earlier, is a specific molecule. Okay, now for this lac operon, the inducer is allolactose, which will inactivate the repressor in an operon by binding to the repressor. Okay, and this repressor here, to be more specific, is the lac repressor. That's what you can see here. Okay, when lactose is present, okay, allolactose from the lactose will bind to this lac repressor and deactivate this lac repressor. By right, this lac repressor is active, always active, because it is being transcribed from the lac-I regulated gene, and this lac repressor should bind to this promoter site. But when this lac repressor is deactivated by the allolactose, okay, the lac repressor cannot bind to this promoter site. So what happened to RNA polymerase? It can transcribe the structural genes, okay, and transcription of these structural genes, okay, we give you one mRNA. Then translation of this mRNA will give you these enzymes. Okay, now this now I mentioned allolactose, okay, came from lactose. So why is that so? Okay, allolactose is an isomer of lactose that is formed in small amount from lactose that enters the cell. So meaning to say, when lactose enters the cell, okay, it will produce allolactose. Okay, lac operon. Okay, let's study the regulation of gene in lac operon. Okay, sorry. Regulation of gene expression in lac operon. Okay, and this lac operon also known as the inducible operon. Now, before we continue here, okay, why you are learning about lac operon? Okay, first things first, it is related with lactose, the presence of lactose. Now, lactose, okay, This regulation gene expression of lactose, okay, is being shown by E. coli, and E. coli are present in your gut, okay, your digestive gut. So now we are going to study how this E. coli inside your body, it regulates the gene expression okay, when you consume lactose. Right, like operon is an operon, okay, that codes enzyme for the transport and metabolism of lactose in E. coli and many other enteric bacteria, gut bacteria. The promoter site of this like operon, okay, has another site for an activated CRP to bind. Okay, now CRP is catabolite regulator protein. Some biologists, they refer this CRP as CAP. Okay, so either it's CRP or CAP, it carries the same meaning. CAP is catabolite activator protein. Okay, and then the CRP or CAP, also known as CAM receptor protein. So meaning to say this CAP or CRP is a protein receptor for CAM. Okay, what is CAM? Cyclic AMP. Right, class. Now in the previous slide, sorry, I mentioned that RNA polymerase binds to promoter, right? It should be operator. So sorry for that. Okay, RNA. So, sorry, class. Let me rephrase that. Oh, okay. RNA polymerase binds to the promoter. Okay, it binds to the promoter. Okay, promoter site. It doesn't bind to the operator. Who binds to the operator? It is the active repressor or active lac repressor. Right? So let me rephrase that again to you. Okay, operator, all right, is a site for the binding of the lac repressor. RNA polymerase binds to the promoter. Okay, and the same goes for the activated CRP. So promoter, it has two sites for binding with activated CRP and RNA polymerase. So these two diagrams here to show you what happens to the CAP, okay, when it is being, how it is being activated and how it is not being activated. Okay, and as I mentioned earlier, okay, the CAP or CRP is a receptor protein for the CAM, and you can see from this diagram here, this CAM, it binds or attached to the inactive CAP. This will activate the CAP, and then the activated CAP will bind to the promoter site. I will explain later on, okay, the detail of this process. Now, in the presence of lactose with low glucose, okay, low glucose, what happens? CAMP accumulates. Okay, so you have lots of CAMP when you drink lactose. When you drink milk, inside milk we have lactose. Okay, but you have not eaten anything yet. So meaning to say your glucose, okay, in your blood is low, right? And then straight away you drink a glass of milk. So what happened? That condition shows presence of lactose with low glucose. So first things first, CAMP accumulates and allolactose binds to the lac repressor. Okay, you can see here, CAMP accumulates and then allolactose will bind to the lac repressor to form allolactose repressor complex, or you can say allolactose bind to the lac repressor to deactivate the repressor, and the deactivated lac repressor cannot bind to the operator. Next, the high level of cyclic AMP or CAM will activate the inactive CAP by attach or binding to the inactive CAP. And then the activated CAP will bind to the lag promoter. Okay, you can see it binds here to the lag promoter. This attachment increases the affinity of RNA polymerase for the lag promoter. RNA polymerase binds to the lag promoter and transcription of structural genes starts to produce large amount of mRNA. The mRNA then translated to produce beta-galactosidase, permease, and transacetylase. Okay, so from here, like Z will code for beta-galactosidase, which will hydrolyze the lactose to glucose and galactose. Like Y will code for permease, which increases the permeability of membrane protein that transports lactose into the cell. So what happens when the cell's membrane, okay, permeability for lactose increases? More lactose will influx, there will be influx of lactose into the cell, body cell of E. coli. Leg A, okay, it calls for transacetylase, which function in lactose metabolism is still unclear until today. Maybe one day you can clear things out for this leg A. Alright. Okay, now I need to mention one thing here. Attachment of cap to the promoter, right, or lag promoter will directly stimulate gene expression. Therefore, this mechanism, okay, is known as a positive regulation. So if lag operon shows positive regulation, okay, attachment of cap to the promoter will show positive regulation, okay, and attachment of cap to the promoter will show a negative regulation. Now, in the presence of lactose with high sugar, what happened? The CAM concentration falls, is the opposite. Okay, and without CAM, the cap detaches from the lag operon. Okay, because cap is inactive, and when cap is inactive, RNA polymerase binds less efficiently to the lag promoter. Remember earlier, when cap is activated, it binds to the lag promoter. This will increase the affinity for RNA polymerase for the lag promoter. Right? Now, when we have high glucose here, okay, the cap is inactive, RNA polymerase will still bind to the promoter, lag promoter, but less efficiently. Transcription of the lag operon proceeds only at a low level, even when lactose is present. Okay. Alright. What about the allolactose? Do they still bind to the lag repressor? Yes, as usual. Okay. Because of that, this RNA polymerase, it can transcribe the structural genes, except that it is being transcribed, except that the structural genes are being transcribed less efficiently. Now, in the absence of lactose, okay, what happens to this lag operon? First, CRP is not activated. Okay, and then the regulatory gene, the lag regulatory gene, okay, here is transcribed into mRNA, as you can see here, that is translated into an active lag repressor. The active lag repressor will bind to the lag operator to deactivate the lag operon, thus prevent the attachment of RNA polymerase to the lag promoter. So no transcription and enzymes of beta-galactosidase, permease, and transacetylase are not produced. Okay, so that's about like operon. Now, why is it a waste to consume milk after a sugar spike in your blood? Okay, so I mean to say the um, digestion of lactose is not properly done. Okay, luckily you have lipase. Sorry, trypsin. Is it trypsin? There's one enzyme in your body that digests the protein of the milk. Renin. Is it renin? Okay. Otherwise, you have some complication because of undigested lactose in your blood. Sorry, in your system. Okay, so now this is the part where you have to answer this abundant of MCQ questions. Okay, so before I reveal out the answers, you pause the video. Okay, so you do this on your own first. I'll give you 5 minutes or 10 minutes. You do all the exercises that's given to you. Okay, all right, see you in 10 minutes time or 15 minutes time. Okay, all right, done. Okay, so to save time, okay, so where I'll give out you the answers. Okay, any part of this question that you not properly understood, please ask us. Right? Okay, all right, okay. Okay, now there's one question here which I didn't write down the answer. When this is taken up by cell, it binds to a repressor, so that the repressor no longer binds to an operator. What is that? Inducer. Right? Inducer. Right? Okay. Alright, 2013 repeat paper. Which statement describes like operand correctly? Right, now class, apparently there are some other questions, past year's questions, which I did not manage to compile here, right? Okay, because of the MCO. Now, please refer to in the internet. I believe they have it circulated, okay, about the past year's questions. Alright, that's it for today. Okay, thank you class. Bye, Michael.